Welcome back to the deep dive. Have you ever noticed how sometimes the smallest, most hidden patterns, well, they're the ones that unlock the biggest secrets? We're talking about those really intricate arrangements that once you understand them, they just totally change your perspective. Today, we're actually applying that exact idea to the human body. I think about it. Our bodies are made of trillions of cells, right? But they don't just float around randomly. No, they organize themselves into these incredibly complex tissues, each with a very specific job. And here's the crucial bit. How those cells arrange themselves, not just, what types of cells are there, is absolutely critical for our health. We're talking everything from fighting cancer to understanding really complex conditions like Alzheimer's. Now, for a long time, our traditional ways of trying to figure out this cellular organization, well, they've kind of missed the forest for the trees, haven't they? Most methods basically group cells by their composition. It's sort of a top-down approach. Imagine you're trying to understand a symphony by just listing the instruments present.
Okay, we have violins, flutes, trumpets.
Yeah.
Know what was there, sure. But you'd have absolutely no idea how they play together or where they sit on stage or the intricate musical patterns they create. You'd completely miss the song's real magic, wouldn't you?
Completely. You lose all the interaction, the structure.
Exactly. And that's where today's Deep Dives comes in. We're introducing a really revolutionary approach called TrimNN. That's TRIMNN. It stands for Triangulation Cellular Community Motif Neural Network. Quite a mouthful.
It is, yeah.
But it represents this game-changing bottom-up strategy. It's designed specifically to uncover those hidden, crucial topological patterns that, frankly, traditional methods just couldn't see. So our mission today is to really unpack TrimNN. What is it? How does it actually work? And what are the profound, sometimes really surprising insights it's already giving us? We'll look at areas like colorectal cancer and Alzheimer's disease. So get ready for, hopefully, a new way to see the fundamental architecture of life itself.
Okay, so to kick us off, the traditional way, just clustering cells by type, it seems logical enough on the surface, but you're saying that's not enough, right? That there's this deeper layer we've been missing. What exactly are the shortcomings? Why isn't it enough to just know what cells are in a piece of tissue? Right. It's a great starting point, but it's only part of the story. What's truly fascinating, and where the field is moving, is understanding that cellular neighborhoods and these larger cellular communities, they're about way more than just identifying the individual cell types present.
Yeah.
It's fundamentally about their spatial arrangement. And crucially, how they interact within that very specific space. So traditional top-down clustering, which groups cells mainly by their overall composition, it has some pretty significant limitations.
Okay, like what?
Well, first, these clustering results can be quite unstable, actually. Picture cells in a really dynamic environment, maybe a developing embryo or a progressing disease like cancer. These cells are constantly changing state. They're migrating, differentiating.
Guys are in flux.
Exactly. And when cells are in those active transitions, the nice, neat clusters we try to draw on paper, they can become really shaky and inconsistent. It gives us kind of a blurry picture.
Right, it's not static.
Not at all. Second, and this brings us back to your symphony analogy, which is perfect. These top-down clusters, they often just describe cell type percentages, but they entirely miss the topological information, the actual geometrical interactions between cells.
Ah, okay, so the where and how connected.
Precisely. Knowing you have, say, 5 red blocks and 3 blue blocks tells you the composition, but it doesn't tell you if they form a sturdy tower or maybe a precarious stack or just a flat mosaic on the floor. The specific shape, those critical connections, they're completely lost in just a percentage count.
That makes sense. You lose the structure.
You lose the structure.
Yeah.
And finally, there's a technical issue, too. These clustering methods can sometimes be overly influenced by something called batch effects. These are basically technical variations between samples, maybe slight differences in staining or imaging rather than true biological differences.
Okay.
So a model might seem accurate because it's accidentally learned these technical quirks in one batch of data.
Oh.
But then it fails dramatically when you try to apply it to new data, maybe from a different lab or a different experiment run. It's like it overfits to the noise, not the biology.
Right, it hasn't learned the real underlying pattern.
Exactly. It's learned the artifacts.
That's a really powerful point about missing the song of the cells. So if just knowing what cells are present isn't enough, what does give us that full, intricate picture? What's the fundamental shift in perspective we need?
That's precisely where cellular community motifs, or CC motifs, become our solution. So instead of that top-down approach, we adopt a bottom-up strategy. Think of these CC motifs as the actual topological building blocks of how multicellular tissues organize themselves.
Okay, building blocks. I like
Yeah,
that.
they're defined as recurring significant interconnections between cells. So these aren't just random groupings that happen by chance. They're conserved spatial patterns, arrangements that we hypothesize are consistent across different samples, maybe different patients, and are directly linked to key biological functions or disease processes.
So repeating patterns of arrangement.
Exactly, recurring significant patterns. And these motifs offer a level of detail that traditional methods just can't touch. They are biologically interpretable. You can actually point to them and understand what they mean in terms of cell types and locations. They give us that crucial topological information beyond just simple clusters, zooming right in on specific cell locations and types.
Okay.
And while, smaller motifs like single cells or pairs of cells, maybe triangles of 3 cells are often studied, what's proving to be truly insightful, especially in recent research, are the sizable CC motifs.
Sizable meaning.
Meaning those with more than 3 cells, so 4, 5, 6 cells arranged in a specific recurring pattern. Biologists are finding these larger motifs significantly correlate with things like patient survival or distinct disease features in conditions like colorectal cancer and various kidney diseases. That's often where the deeper biological truths seem to emerge.
Wow. Okay. So moving from just individual notes or simple pairs to understanding these larger chords and like melodic phrases.
That's a great way to put it, yes.
But identifying these intricate patterns, especially the larger ones you're talking about, that must be a computational nightmare, right? It sounds incredibly complex, like trying to find every unique snowflake in a blizzard.
You've absolutely hit on the core computational hurdle there. Finding these overrepresented CC motifs, especially as they get larger, is indeed extremely expensive computationally. It's a huge challenge. Classical methods for what's called subgraph matching, which is basically how we count the occurrences of a given pattern or motif within a larger network or graph.
Right.
They are just too slow, they scale very poorly, they quickly become impractical for the kinds of large data sets we're generating today with spatial omics technologies. We're talking thousands, sometimes millions of cells.
So the traditional tools just can't keep up.
They really can't. Imagine trying to manually check every single possible combination of, say, 5 cells in a tissue with 10,000 cells. It's virtually impossible at scale. And this challenge has historically limited our ability to fully leverage all the rich information in this spatial data.
Okay, so if the traditional computational methods basically hit a brick wall, how does TrimNN, this new player we're discussing, manage to tackle this seemingly impossible problem? What makes it the game changer here?
Right. This is where TrimNN truly shines. It's a graph-based deep learning framework. It's specifically engineered to efficiently and effectively identify these conserved spatial cell organization patterns, or CC motifs. It employs that bottom-up strategy we talked about, building from local interactions.
Right.
But it does so with a really revolutionary computational approach. TrimNN's core mission, you could say, is to provide a computationally feasible way to identify these conservative, interpretable, and generalizable spatial rules of cellular organization. And it does this across different motif sizes, small and large, and across many different biological samples. It essentially transforms what was an impossibly slow computational task into something manageable, often in mere seconds for typical data sets.
Seconds. That is quite the claim. Seconds instead of hours or potentially days with the old method.
It's a dramatic speed up, yes.
Okay, before we dive into the technical how of TrimNN, let's just quickly reinforce why this all matters so much. Quick recap. We've established that understanding how cells organize themselves in space is absolutely crucial for health and disease. It's not just what cells, but how they're arranged.
Correct.
And that traditional clustering often misses this vital topological recipe for cell interactions. It struggles with dynamism, with noise, batch effects.
All true.
Now with CC motifs, we're looking at these fundamental, interpretable topological building blocks. And especially the sizable ones, those with more than 3 cells, seem key to gaining deeper insights.
Yes, the larger motifs are proving very informative.
And TrimNN is the engine, the new tool, that finally makes searching for these complex patterns computationally feasible.
Precisely.
It's like we're moving from just having a simple list of ingredients to finally understanding the whole intricate cooking process, the recipe itself.
That's a fantastic analogy, yes.
Okay, so now for the how. How does TrimNN actually see these hidden connections? What's going on inside this system, this deep learning framework, that allows it to piece together these subtle spatial patterns so effectively?
Okay, let's break it down. It all starts, naturally, with the raw data. TrimNN takes as input what we call spatial omics data. These are generated by really advanced technologies. You might have heard names like StarMap Plus or Codex or Mibitov.
Right, those high-plex imaging methods.
Exactly. They give us essentially a high-resolution map of a tissue sample, showing the precise location and the specific type, like immune cell type A, tumor cell type B, of every single individual cell.
So a detailed cellular map.
A very detailed map. From this map, TrimNN then constructs what we call a cellular community, or a CC graph. Think of each individual cell on that map as becoming a node in a network, like a dot, and its specific cell type becomes the node type or label on that dot.
Okay, cells are dots, types are labels. Makes sense.
Now, the crucial step. How do we define who's truly a neighbor? How do we draw the connections, the edges between these nodes? TrimNN needs to connect cells that are physically close, that are actually interacting. It does this using a rather clever geometric trick called Delaunay triangulation.
Delaunay triangulation. Sounds complex.
It sounds complex, but the concept is fairly intuitive. Imagine you scatter a bunch of points on a map representing your cells. Delaunay triangulation connects these points to form triangles, but it does so in a very specific way. It ensures that if you draw a circle around any single triangle, no other point, no other cell falls inside that circle.
Ah, okay. So it finds the most direct neighbors.
It's a very robust way to define immediate neighbors based on proximity, creating a geometrically sound graph that represents the cellular interaction network.
Got it. So now we have a graph, nodes, types, and edges based on proximity. What's next? How does it find the patterns?
Right. Now we need the pattern recognition engine. The core mechanism involves advanced deep learning, specifically a type of model called graph isomorphism networks, or GNs. GNs are particularly powerful for learning complex relationships and patterns within graph structures, like our cellular community graph.
Okay, GINs. What do they do, essentially?
Think of a GIN as a special kind of AI that can look at two different networks, maybe two different cellular communities from two different patients, and determine if they share the same underlying structural pattern, even if they're drawn slightly differently or have different numbers of nodes overall. It's a bit like recognizing the same melody, whether it's played on a piano or a guitar. The instruments, the specific notes might differ slightly, but the underlying pattern, the relationship between the notes, is the same. GNs are good at capturing that pattern.
Okay, pattern matching for graphs.
But here's a really crucial enhancement that GIN uses. It integrates something called positional encoding, or PD. A standard GIN might know who a cell's immediate neighbors are based on the edges. But PE adds vital spatial context. It tells the network where a specific cell is relative to other cells, maybe cells that aren't immediate neighbors, often using measures like the shortest path distance between them on the graph.
Ah, so it's not just direct contact, but the broader neighborhood layout.
Exactly. It's the difference between knowing everyone who's standing right next to you in a room versus also knowing how far away other people are and their general positions relative to you. That broader spatial context is absolutely key for understanding tissue architecture and biological function.
That makes a lot of sense. So GINs for pattern, PE for spatial context. Now, how does it handle that speed issue, the computational hurdle?
Right, back to the speed. This is where TrimNN uses an ingenious strategy. They call it a semi-divide and conquer approach. Remember we said finding entire complex patterns like large motifs all at once, subgraph matching, is computationally explosive, practically impossible for large graphs.
Yeah, the NP complete problem.
Exactly. So instead of tackling that head-on, TrimNN braced down this immense problem into many, many smaller, much more manageable binary classification tasks. Essentially, for each node, each cell, it looks at a small, sort of enclosed graph region right around it.
Like a little local neighborhood?
A little local neighborhood, yes. And within that tiny neighborhood, it simply predicts the presence or absence of a specific, smaller motif. Is this little triangle pattern here? Yes or no? Is this little square pattern here? Yes or no? Much faster to check locally.
Okay, lots of small checks instead of one giant one.
Precisely. And then it combines this with a pattern growth strategy. It starts by identifying occurrences of small, simple motifs, like pairs or triangles, using this classification approach. And then it incrementally searches for larger motifs by seeing if these smaller ones connect together in specific ways to form bigger successor motifs. It builds up complexity gradually.
Clever, sort of growing the patterns from smaller seeds.
That's a good way to think about it. This combination makes finding even quite intricate motifs computationally feasible, bringing it down to those seconds we talked about.
Okay, so the approach sounds really smart. is breaking down the problem, adding spatial context. But does this sophisticated approach actually work better in practice? Does it deliver real, measurable improvements compared to what we had before?
Oh, absolutely. The performance gains are quite significant across several key metrics. First, its robustness to noise is really remarkable. Real-world spatial data is never perfect, right? You might have some missing cells, slight shifts in the cell coordinates, maybe even some minor misclassifications of cell types.
Sure, data is messy.
It is. TrimNN handles these common imperfections surprisingly well. Even with relatively high levels of simulated noise introduced into the data, it maintains surprisingly consistent rankings of which motifs are most important or abundant.
So it doesn't get thrown off easily.
Not easily. It's like a skilled detective who can still piece together the pattern of a crime even if some pieces of evidence are smudged. Or maybe one witness account is slightly off. It sees through the noise to the underlying structure.
That's impressive. What about accuracy and speed?
On accuracy, it consistently outperforms previous methods, particularly in identifying those larger, more complex motifs, the ones that are often more biologically revealing. Methods like NSIC or the classic VF2 algorithm just struggle with those larger patterns. And then there's scalability. This is arguably the biggest leap for practical, real-world applications. TrimNN exhibits linear scalability with increasing graph size, meaning the computation time grows roughly in proportion to the number of cells.
Linear, not exponential.
Exactly. Contrast that with older classical methods like VF2, where the runtime grows exponentially. They quickly become practically unusable for the size of modern spatial omics datasets, which can easily have tens or hundreds of thousands of cells. TrimNN completes a comprehensive analysis in seconds or maybe minutes for these datasets. That combination of the GIN architecture and the positional encoding really enhances its expressive power, its fundamental ability to understand and differentiate these complex spatial relationships efficiently.
Okay, that's fantastic to hear about the performance and benchmarks, speed, accuracy, robustness. But let's ground this in reality. How does this translate to actual biological insight? Say, in understanding a disease as complex as colorectal cancer. What did TrimNN actually reveal there?
Yeah, let's look at a really powerful case study in colorectal cancer, or CRC. The researchers applied TrimNN to analyze data from a spatial proteomics study using that codex technology we mentioned earlier. They compared tissue samples from 17 patients classified as low risk, CLR, with samples from 18 patients classified as high risk, DII.
Okay, low risk versus high risk CRC.
Right. And the first key finding was TrimNN's superior classification ability. They took the CC motifs identified by TrimNN, these spatial patterns, and used them simply as features in a relatively straightforward machine learning model, a logistic regression model.
Okay, a standard model.
A standard interpretable model. And just using these motif features, it dramatically outperformed more complex, black box deep learning approaches that others had tried on the same data for classifying patient risk. For instance, using just 100 features derived from size 3 motifs, TrimNN's patterns achieved an ROC-AUC score, a measure of classification accuracy of up to 0.81.
Which is pretty good.
Very good. Compared to about 0.71 for a competing deep learning method. This tells us that the specific spatial patterns TrimNN found were incredibly distinguishing between the low-risk and high-risk groups. They captured something biologically fundamental.
Okay, better classification is great. But you mentioned something even more profound earlier about survival.
Yes, and this is where it gets really critical. Sizable motifs predict survival. This was a striking finding. They found that while simply knowing the overall cell type composition, which you could think of as size 1 motifs, just the individual cell types present couldn't reliably differentiate patient survival rates within that high-risk group. The sizable CC motifs, identified by TrimNN, specifically size 3 and size 4 patterns, showed significant links to lower survival rates for those high-risk patients.
Can you give an example?
Sure. Let's say we have a type of immune cell, CD68 plus CD163 plus macrophages. Let's call them type A, and smooth muscle cells, call them type B. A simple pair motif, just A next to B, a size 2 motif, didn't really separate the survival curves well. The difference wasn't statistically significant.
Okay.
But when TrimNN identified specific sizable motifs, like the size 3 pattern A and D, 2 macrophages next to a smooth muscle cell in a triangle, or the size 4 pattern A and B, 2 macrophages interacting with 2 smooth muscle cells, Patients whose tumors showed an enrichment of these specific, larger topological arrangements had significantly lower survival rates. The p-values were highly significant, 0.016 and 0.0093 respectively.
Wow, so the specific arrangement of 3 or 4 cells was predictive, or just the pair wasn't?
Exactly. It demonstrates powerfully that it's not just what cells are nearby, but their precise geometrical arrangement in these slightly larger structures that can directly impact patient outcome. It's a much deeper level of biological information.
And was this interpretable? Could they see why these patterns mattered?
Yes, and that's another strength. Because they used that simple logistic regression model, it was intrinsically interpretable. The model assigned high importance, high absolute coefficients precisely to those macrophage-related and muscle-related motifs. This wasn't just a black box spitting out a prediction. It directly highlighted the biological relevance of these specific cellular interactions. And importantly, this validated known biological crosstalk in cancer, where certain macrophages are known to interact with muscle cells, potentially facilitating muscle wasting, or cachexia, which is linked to poor prognosis.
So the math confirmed the known biology and even refined it by showing the importance of specific spatial configurations.
Precisely. It provided spatial context to known interactions.
That's a really powerful result. Going from just classifying disease risk to actually predicting patient survival based on these detailed spatial patterns. It truly highlights that these cellular recipes, these CC motifs, are vitally important.
Absolutely. It opens up new avenues for thinking about biomarkers and potentially even therapies targeting these interactions.
Okay, let's take our second quick recap here. We've just heard how TrimNN works its magic. It builds these cellular community graphs using Delaunay triangulation cells as nodes, proximity as edges.
Right.
It then uses smart deep learning GNs with that crucial positional encoding, plus a divide and conquer approach to efficiently and accurately find these CC motifs.
Yeah, breaking down the problem.
We learned it's highly robust to data noise, scales linearly, which is huge, dramatically outperforming older methods in speed and accuracy, especially for larger motifs.
Key advantages, yes.
And in that colorectal cancer example, TrimNN identified motifs, particularly sizable ones like that A and A and D pattern, effectively classified patient risk and predicted survival, something simple composition couldn't do. Plus, it was interpretable.
A very compelling case study.
Okay, thinking about this, consider the difference between just knowing the ingredients for a cake and actually having the step-by-step recipe that tells you how to combine them. How might understanding these detailed recipes of cell interactions actually change how we approach developing new disease treatments in the future?
That's the exciting frontier, isn't it?
It really is. Now, moving beyond cancer, which is obviously huge, TrimNN is also shedding light on incredibly complex neurological conditions, Alzheimer's disease, for instance. What insights did it uncover there, especially looking at that interplay between microglia, the brain's immune cells, and neurons?
Right. Alzheimer's is another area where understanding spatial context is proving critical. In a study looking at an Alzheimer's disease mouse model using that StarMap plus S transcriptomics technology, TrimNN was used to identify significant CC motifs. They focused particularly on interactions involving CTX excitatory neurons, let's call them C, and microglia, M. It found very significant size 3 motifs like CCC, 3 neurons together, CCM, 2 neurons, 1 microglia, CMM, 1 neuron, 2 microglia, and even MMM, 3 microglia clustered.
So different local combinations of these key cell types.
Exactly. And the first key finding was simply the uneven spatial distribution of these motifs. They found that these specific patterns showed strikingly different arrangements and abundances in the AD brain samples compared to the healthy control samples. The cellular landscape was clearly altered at this motif level.
Okay, the patterns were different. What else?
Well, beyond just where the motifs were, they looked inside these motif-enriched regions. They found significant differences in ligand receptor signaling pathways between AD and control samples within these specific motifs. We're talking about communication pathways known to be involved in AD using ligands like VDGF and PDGF. This clearly points to distinct cell-cell communication patterns happening within these specific structural arrangements in the diseased brain.
So the local signaling is different within those patterns.
Precisely. It also revealed gene-level insights. When they looked at gene expression within the cells forming these motifs, they found differentially expressed genes turned up or down like AP, amyloid precursor protein, CLU, clusterin, and TREM2. These are all genes well known to be involved in Alzheimer's pathology, linked to amyloid formation and the risk of late-onset AD. So TrimNN directly connected these specific spatial patterns to the underlying molecular changes of the disease.
Wow, connecting structure to function and genes.
Exactly. And perhaps one of the most striking findings was related to the infamous amyloid plaques. TrimNN showed distinct patterns of co-localization. Specifically, microglia-related motifs like MMM or even size 4 MMM motifs had a higher probability of being found spatially close to these amyloid plaques.
Okay, microglia clustering near plaques. That makes sense biologically.
It does. They're the immune responders. Conversely, the CTX excitatory neuron-related motifs, like CCC, showed litter co-occurrence, meaning they were less likely to be found right next to the plaques.
Suggesting maybe damage or avoidance?
Exactly. It suggests potentially two distinct types of microenvironments related to plaques. One characterized by microglia actively engaging near the plaques, possibly driving inflammation, and another where neurons might be more distant, perhaps damaged or cleared, or simply not part of that immediate plaque interaction zone. These kinds of insights really help us refine our understanding of the cellular dynamics in Alzheimer's progression.
That's incredibly detailed. Okay, and let's touch on one more fascinating example. Back to colorectal carcinoma, but looking specifically at the immune environment dynamics. It sounds like TrimNN can pick out some very specific shifts in how immune cells interact with tumor cells or even amongst themselves.
Indeed. The tumor microenvironment is this incredibly complex ecosystem, and TrimNN provides a new way to dissect it. In another spatial proteomic study of colorectal carcinoma, TrimNN identified two really novel types of CC motifs that reveal these dynamic changes. The first type they called shifted interaction motifs. A good example was seeing a size 3 motif, say involving CD4 T cells, CD8 T cells, and epithelial tumor cells, let's call it ABC,
Right.
transitioning into a size 4 motif, ABCD, by adding perhaps another type of immune cell, maybe a macrophage or a regulatory T cell. Crucially, this larger expanded motif, ABCD, showed significantly increased abundance in the actual disease samples compared to nearby normal control tissue.
So the interaction pattern expanded in the tumor. What did that mean biologically?
Well, when they looked at the protein expression within these shifted motifs, it pointed towards heightened but potentially dysfunctional immune activity. They saw upregulation of proteins like CD39, which is associated with immunosuppression, and various metabolic enzymes. This suggests that the structural shift, this expansion of the motif, corresponds to the tumor niche becoming more immune suppressive and metabolically challenging, likely helping the tumor to evade the immune system.
So the changing structure reflects a changing, more suppressive function.
Exactly. Now, the second type they found was homeostatic interaction motifs. This was interesting because here, the basic structural motif, for example, involving CD4 T cells, endothelial cells from blood vessels, and epithelial cells, AEC, remain pretty consistent in abundance between the disease and control areas, even when it expanded slightly to, say, AECC.
So the structure looks stable.
The relative abundance of the motif structure seems stable, yes. However, when they looked at the protein expression within the cells forming these stable motifs, it had changed dramatically in the tumor environment. They observed significant upregulation of proteins like PD-1, an immune checkpoint marker indicating T-cell exhaustion, and GLUT1, involved in glucose metabolism, suggesting metabolic competition.
I see. So even though the overall arrangement pattern stayed the same, the state of the cells within that pattern was changing.
Precisely. It tells us that even within stable structural contexts, the underlying biology is shifting significantly in the tumor microenvironment. Immune cells are becoming exhausted, or they're competing for resources. So taken together, these shifted and homeostatic motifs revealed by TrimNN show how structural reorganization or functional changes within stable structures correspond to distinct biological states. They point directly towards mechanisms of tumor progression and immune evasion.
That's fascinating. It really paints a dynamic picture. So stepping back a bit, TrimNN isn't just another tool in the toolbox, is it? It seems like it's fundamentally changing how we look at tissue biology, offering these incredibly detailed insights into disease mechanisms. Why is this such a big deal for the broader scientific community?
I think it is a really big deal for a few key reasons. First, it provides crucial complementary power to the existing methods we have. It doesn't necessarily replace top-down clustering altogether. That still gives you a useful, broad overview. But TrimNN fills a critical gap by offering this fine-grained, bottom-up perspective focused on topology and local interactions.
So it adds a different essential layer of information.
Exactly. Think of it like maybe understanding a complex protein. Clustering might tell you about the large functional domains, while TrimNN or motif analysis tells you about the critical short sequence motifs within those domains that actually determine binding or activity. You really need both levels.
That's a great analogy.
Second, it offers intrinsic interpretability. As we saw in the examples, the results, these CC motifs, are generally easy to understand at the cellular level, the gene level, and even connecting to pathology. You're not just getting abstract numbers or embeddings from a complex model, you're getting identifiable patterns that directly link back to known biology, which builds confidence and facilitates new hypotheses.
Right, it's not just a black box.
Much less so. This interpretability also leads to better generalizability. Because the features are biologically grounded, models built on them are potentially less prone to overfitting to specific dataset quirks compared to some highly complex models that learn very abstract representations. And ultimately, all of this opens up really exciting new opportunities for understanding fundamental processes like multicellular differentiation during development or tissue regeneration and certainly disease progression. It could help us, for instance, to better identify and characterize different types of tumor microenvironments, distinguishing cold tumors with few immune cells from hot ones, or identifying mixed or compartmentalized tumors, which we know respond very differently to immunotherapies.
So potentially leading to more personalized medicine based on the spatial architecture.
That's definitely the hope, yes.
Now, even the most powerful tools usually have some areas where they could grow or limitations to be aware of. What are TrimNN's current limitations? And what's next for this technology? Where does the research go from here?
That's a very important question. No tool is perfect, and TrimNN is still evolving. Currently, its search strategy for finding those large motifs relies on what's called a greedy search. It works well, but there's potential room for improvement, maybe using more advanced techniques like deep reinforcement learning to explore the search space even more effectively.
Okay, refining the search.
Right. Also, while it gives excellent relative rankings of motif abundance, telling you motif X is more common than motif Y, it doesn't always guarantee mathematically exact counts of every single motif instance, which might be needed for some very specific statistical analyses. That's another area for potential refinement.
Makes sense.
And for now, the way it builds those cellular community graphs is a bit simplified. It mainly considers cell type and proximity. Future versions could potentially incorporate more features directly on the edges, connecting cells like the length or type of interaction, or on the nodes themselves, like cell shape or morphology, which could add even richer context.
Adding more layers of information.
Exactly. So future work will definitely involve applying it to even larger and more diverse data sets, developing perhaps better statistical metrics for evaluating the significance and optimal size of motifs, and of course exploring its applications across a much wider range of biological systems and disease categories beyond CRC and AD. And related to that, there's this fascinating ongoing discussion and challenge about selecting the optimal motif size for any given question.
Oh, right. How big should the patterns be?
Yeah. While larger motifs, as we've seen, can offer potentially better predictive power and capture more complex interactions, they are inherently harder to identify statistically. You need more data to be confident they aren't just random chance occurrences. They often lack well-established statistical frameworks compared to smaller motifs, and there's always a higher risk of overfitting your model if you use very complex features with limited data. So finding that sweet spot, the motif size that gives you the most robust and meaningful biological insight for your specific context, is an active and really interesting area of ongoing research.
A balancing act, then.
Very much so.
Well, this has been an absolutely incredible deep dive. We've really seen how TrimNN, by shifting the focus to the intricate spatial topology of cells, not just what cells are present, but how they're specifically arranged and interact, is truly revolutionizing our understanding of tissue biology and disease. It really is like we've gone from just listing the instruments in the orchestra to finally hearing the full, complex, beautiful symphony they create together.
That captures it perfectly. It offers an entirely new lens, a new perspective, showing us that the how of cellular arrangement is just as critical, maybe even more critical in some cases, than the what. These hidden connections, these motifs, they really are the true language of tissue biology.
So the final takeaway here seems to be, TrimNN provides this unprecedented spectrum of spatial relationships. It reveals these subtle, yet often profound differences in tissue architecture that were often overlooked by conventional methods just looking at cell counts or broad clusters. And these cellular community motifs, they aren't just interesting patterns. They look like they could be powerful diagnostic biomarkers, crucial clues to how diseases progress, and maybe even represent entirely new types of therapeutic targets.
That's the potential, absolutely.
So, a final thought to leave with everyone listening. As we gain this incredible new ability to effectively read the language of cellular communities with such precision, using tools like TrimNN, What groundbreaking biological and medical discoveries do you think are just around the corner? How might this deeper understanding of spatial organization fundamentally reshape our approach to medicine entirely, maybe moving us closer than ever to truly personalized and effective treatments based on the unique cellular architecture of each individual's tissues? Something to ponder. Thank you so much for joining us on this deep dive into TrimNN and the absolutely fascinating world of cellular community motifs.
My pleasure. It was great discussing it.
We'll see you next time on The Deep Dive.